To everyone. This is Margaret Kanamuri, Head and Assistant Professor, Department of Biotechnology, St. Joseph College of Arts and Science for Women, Hosur. In this video, we are going to see about the structural level organization of proteins. Proteins are made up of amino acids. The amino acids are the building blocks of the proteins. Several amino acids join together by means of peptide bonds and it is producing the proteins. In the structural level organization of proteins, there are four types of them: primary structure, secondary structure, tertiary and quaternary structure. In primary structure, the proteins refers to the sequence and arrangement of amino acids in the polypeptide chains. Alpha carboxyl group of one amino acid is linked with the alpha amino group of the other amino acids by means of this peptide bond. The successive peptide bonds linked amino acids, it will produce the proteins. In polypeptide chain, alpha carboxylic group and alpha amino group of this amino acids are involved in the formation of this polypeptide. The two amino acids are present at the either end of the polypeptide chain, have the free carboxylic group and free amino group. So this carboxylic group is free, means that is called the C terminal N, and which the amino group is free is called the N terminal polypeptide chain. The alpha carboxylic and amino group formed peptide bonds, they are not available for forming the bonding with other component. Here it is forming the hydrogen bondings. Next we will move on to the secondary structure of these proteins. The amino acids which are located near to each other interacts with each other and it form a regular arrangement is called the secondary structure. The secondary structure is involved the local folding of the polypeptide bond. There are three commonly occurring secondary structures are there: alpha helix, beta sheet, beta bends or beta turn. Alpha helix structure. This alpha helix structure of the protein is the right-handed helical structure. It is formed by twisting of the polypeptide chain. It is a spiral structure. Each helix in the alpha helical structure, it contains the 3.6 amino acid residues. The vertical length of each helix is known as the pitch. That Armstrong is 5.4 Armstrongs. The vertical distance between the two nearest amino acids is 1.5 Armstrong. So this is called the identity period of alpha helix. In alpha helix, the carboxyl group of each amino acid is hydrogen bonded with amino group of other amino acid which is present near to the 4 amino acid ahead. Therefore, the carboxylic and amino group of all amino acids are hydrogen bonded in the alpha helical structure. The R group of amino acids in alpha helix is always projected outward to minimize the steric hindrance. Some amino acids, it will disturb the alpha helix. For example, the amino acids are charged with R group. It will disturb the alpha helix by means of electrostatic repulsion or by the formation of the ionic bond. Similarly, amino acids with bulky R group, that is also disturb the alpha helix by steric interference. The amino acids glycine and proline, it will bend in polypeptide chain and disturb the alpha helix. Next we will move on to beta sheet structure. This is also comes under the secondary structure of this protein. This is the most stable form of secondary structure of proteins. It is formed between two different polypeptide chains. These two different polypeptide chains which are placed parallel or anti-parallel to each other. It can be formed by folding of same polypeptide chains. The beta sheet structure, the two polypeptide backbone are linked with each other by hydrogen bond which are formed between of carboxylic and amino group. The R group of this amino acids, it is projected above and below the plane of the beta sheets. This surface of the beta sheets is not straight, but it is pileated. That's why it is called the beta pileated sheets. The polypeptide backbone in beta sheet is extended rather than being tightly coiled as in alpha helix. The axial distance between of two nearest amino acids is 3.5 Armstrong and 1.5 Armstrong in alpha helix. The beta sheet hydrogen bond may be interchain or intrachain, but always they are in the interchain alpha helix. Beta bends or beta turn structure. This beta bend reverses the direction of polypeptide chain, and it helps with the formation of a spherical structure. It consists of four amino acids. Glycine and proline are always more found in beta bend structures. The ring of proline is attached with the alpha carbon atoms so that it will helps to bend the chain, and R group in glycine, it will give the degree of rotation around the alpha carbon atom and bring bend in polypeptide chain. Tertiary structure of proteins. This is the overall folding of the polypeptide chain, and it will give the three-dimensional structure. For example, globular protein. This is the larger form of the protein. It contains nearly 200 amino acid units. It forms two or more domains by folding of polypeptide chain by either alpha helix or beta pileated sheets or beta bend. And these domains are associated with each other to form a final 3D structure. So the tertiary structure refers to the formation of these domains, and it is giving the final folding of this protein structure, and it will gives the 3D structure of this globular proteins. This bonds for the formation of this tertiary structure, the bonds like hydrogen bond, hydrophobic interaction, ionic bond and disulfide bonds, it is very helpful in holding of this polypeptide chain during the formation of this tertiary structure. This picture indicates what are the bonds are involving in the formation of tertiary structure. Ionic bond, polypeptide backbone, hydrophobic interactions, disulfide bond and hydrogen bonds. All these bonds are joined together, and it is helpful for the formation of this tertiary structure. Quaternary structure of proteins. Some proteins are composed of more than one polypeptide chain, and this polypeptide chain in such proteins are called the subunits. This is the very compact manner of this structure of the protein. This quaternary structure, it's like interaction between of the subunits to form the final 3D structure of this proteins. So this quaternary structure refers to this interaction between different polypeptide chains of multi-chain proteins. We can found only in the protein which are composed of more than one polypeptide chains like hemoglobins. The bonds which are involved in this quaternary structures are hydrogen bond, ionic bond, hydrophobic interactions. These bonds are helpful for the formation of this quaternary structure. And this is the pictorial representation of this protein structure. Here this amino acids, the bead-like structure of this amino acids are joined together by means of the peptide bond, and it is producing the proteins. And this is the primary protein structure. And here if you are seeing the secondary protein structure, here there is not a smooth surface of the secondary structure. That's why it is called the pileated sheets. Here the hydrogen bond of this peptide backbone, it will causes the amino acids to fold into the repeating pattern. We will get this. Tertiary protein structure. This is the three-dimensional folding pattern of the proteins due to the side chain interactions. Quaternary structure of this protein, it is consisting of more than one amino acid side chain. Assessment part. You can, dear viewers, you can give the answers in your comment box. Thank you for watching this video.